From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 13, recorded on December 14, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about the nervous system. Joining me today from New York, Ori Lieberman.
Hi, Vincent. How are you?
I am well. We are, did you get some snow there? Some flurries today?
So I have a secret to tell you. I'm actually in California.
That's right. You're not. I look out your window and I don't see.
It's light outside.
It looks a little bright for New York.
Nice.
Yeah.
Very good.
I'm enjoying the sun out here doing Zoom interviews for residency. So
are you north, middle, or south?
I'm in Sacramento, which they tell me is north, but on the map it looks kind of in the middle.
Oh, so. Cool. Well, my video guy from ASM, Ray Ortega, is in Sacramento.
Oh.
Very cool. Also joining us from Salt Lake City, Jason Shepard.
Hi, Vincent. How's it going?
Hi, Jason.
How are you? Everything good?
Yeah, pretty good. Actually feels like winter finally, so we've got some snow.
All right. And from Nashville, Tennessee, Erin Calipari.
Hi. We actually got snow too, by the way. Like one flurry one day, but it's been like 75 since then. So that's our winter down
75?
here. It's quite warm. So
you've been away for a few times. Good to see you back. Did you get all your grants done?
I did. I actually got a good score on one of them, so I can't complain. So it's a good day to be back. And now it's the holidays, so things are a little bit more, I'd say maybe less chaotic, but I don't know if that's true. It's a different kind of chaotic. So
today, of course, first day that the U.S. is immunizing people against COVID-19. It's amazing. It's been less than a year. I'm stunned, frankly.
It's crazy how fast it went. I mean, now we get to look at the logistics and distribution and how effective that is. But the first step of this went really, really fast.
And it worked. I mean, that's the amazing part. It's one thing to plan different vaccines and try them, but they all seem to go pretty smoothly. And as some of you may know, clinical trials are no day at the beach, right?
Yeah, so amazing. I think that is 95%. I think that was the cherry on the top and how well they seem to work.
Yeah.
And then the second surge also helped.
Yes, we got the results quickly.
Yeah.
You remember for a while we were talking about challenge experiments because we were worried there weren't going to be enough cases, right?
Yeah.
But that wasn't a problem. So, and we have more vaccines to come. There are a couple, 2 or 3 more in the U.S. at least, pretty far advanced. So could be interesting.
Well, there's the Moderna one, like the meetings with the FDA are this week, right? So
that's right, 17th,
we
right?
get to have another approval.
Yeah, and then we have AstraZeneca supposedly, and then Johnson & Johnson and Novavax are not too far behind.
Wow,
and I
pretty
think
impressive.
It's just amazing, really. Back in February, I thought maybe next fall we'd have some vaccines, but here we go. Faith in science, that's what it is. It's all science.
That's right.
A lot of good things ending up. All right, but we're not going to talk about viruses today. We're going to talk about why I can't remember things for long periods of time. Apparently it has something to do.
To our favorite, well, at least my favorite subject.
And I think Jason's going to lead us through this, right?
That's right. So this is a paper that was published in Nature a month ago called Persistent Transcriptional Programs Are Associated with Remote Memory. And this is from a group at Stanford. This is Tom Sudoff and Stephen Quake. Sudoff won the Nobel Prize a few years ago for his work looking at synaptic transmission. He did a lot of real groundbreaking work looking at how neurotransmitter is released in synaptic vesicles from synapses. So he's a big lab. So this sort of introduces a couple of new-ish terms that I just wanted to start off with, where I think last time we talked about memory, we talked about how there's the structure called the hippocampus that's required for making new memories. And then there's this process called consolidation that can then convert short-term information into long-term storage. And there's this idea that the hippocampus is required initially to start off the cascade of consolidation, but the ultimate site of where information is stored has been somewhat hard to figure out, and in some ways has been controversial. So going back to like the old days where there was two camps. One camp is that the information in the brain is distributed across different parts of the brain, that there's no like one site for a memory. You couldn't go in and say, this is where the memory is stored. So that was the one extreme. The other extreme was that, no, there's actual cells that are essential for each memory. And if we just knew where those cells were, we could then reconstitute the memory by just activating those particular cells. So there was back and forth, and I would say that the modern day hypothesis, or the modern day way of thinking, is that it's more on the circuit side, that there are specific cells for each memory, but it's still not clear where those cells are.
Right. What components of memory each cell makes up. So it becomes even more complicated, right? Because something that's too good experiences, there may be overlapping in the cells that are recruited by positive things, but then there's specific aspects. So it's such a complicated process that we're getting tools to parse apart now, which is why I'm excited about this particular paper.
Yeah, exactly. So this paper is, it brings in a lot of the brand new technology that we have for figuring out a few aspects of this memory consolidation process. So that's one sort of new concept here, that the cortex and then parts of the cortex we think end up being where the information is ultimately stored, and that the hippocampus you don't actually need anymore after this consolidation process. So this is called systems consolidation, where the information is ultimately consolidated in a new part of the brain, not the hippocampus, which is where the initial information is processed. And the other aspect of this is that, when we think about consolidation of memory, and we think about, well, I guess the lay public thinks about memory is that, it's some sort of videotape of our experiences. And of course, that's completely false, because we end up changing the memory every time we recall it. So there's subtle updates to the memory literally every time you recall it. And so there's this other concept called reconsolidation, which is that when you recall the memory and there's an updating process that happens, that memory can be wiped out if you interfere with it right at that point. So it becomes this sort of more plastic thing, and then it gets reconsolidated, and now it's now consolidated again into the brain, but with a slight update. And so if you can think about it, every time you recall a memory, by the 10th time you record the same memory, it's absolutely not the same as the first time.
But this is a really cool process to understand from just a basic neuroscience. How does the brain work? But also, how do we treat disease, right? Because if you think about things like PTSD, where you have these kind of really strong, recurring, kind of inappropriate memories that trigger anxiety responses, how can we use what we know about kind of retrieval and reconsolidation to target those memories and get rid of certain memories that are intrusive while retaining the memories that are really important for our navigation of our environment? And so these studies are so important for just how does the brain work? but also how can we kind of harness this to really treat this in disease, but also not have adverse effects. So that's why this is so exciting, right? There's just so many levels for, if we can figure this out, how we could use it in disease and all kinds of things like that.
Yeah, exactly. So one idea is that PTSD is this, super salient memory that's tied to an emotional content. And perhaps one way to get rid of the emotional side of that memory is to have people recall the memory in a safe context, a safe environment, so that over time that memory gets updated, but you sort of start to lose the real strong emotional content. And that has panned out in human studies, where some of the therapies that people use for PTSD are exactly that you sort of get people to recall the really painful memory, but in a very safe context, in a safe environment.
Jason, in the humans, how long does it take for this memory consolidation? And does it require, so the original thing that you're remembering, do you have to repeat it, or is it enough to do it once?
Yeah, those are all good questions. So there are some kinds of memories that you only need one event. So, and that's usually these emotional kinds of memories where you can imagine that they stick in your mind, partly because they elicit such a big emotional response. For other things that are boring as hell, like trying to memorize, math equations, you do need repetition. So the brain is wired to consolidate things that are going to elicit an emotional response, because, of course, in the environment there are these stressful things that you would want to learn quickly are things like, where's that predator, or I ate that bad food and I don't want to eat that again.
It's all about survival, right?
Right. Exactly. And in terms of the timing, it's not actually that clear, but I would say, generally speaking, this molecular consolidation happens within days after learning. But the systems consolidation, where the ultimate storage in the cortex happens, takes about 3 to 4 weeks, so about a month. And so in this study, and again, this is a study in animals, so they're using mice. Their setup is their behavioral setup is this fear conditioning memory. So they basically train the mouse with putting them in a specific context in a box, and they shock the mouse, a mild shock, enough that the mouse obviously doesn't like it. And then a couple of trials, it doesn't take that long, because this is the kind of memory that does elicit emotion in a big response. The training is pretty short, so they only take a day for training, and then you can get a robust memory that lasts for months. And the consolidation, so then they basically take the animal after learning, leave them alone for 2 weeks, 16 days, and then put them back in that same box that they were shocked in, where they know that the animal is going to recall that memory. And at that day, 16, they use some fancy genetics in the mouse to mark the neurons that are active during that recall event, with the idea that those active neurons that are being activated during the recall event are the neurons that are part of the consolidation process, or the reconsolidation process. And then they come back 9 days after that recall event and actually take out those cells. So they've used the mouse genetics to express a fluorescent protein in those active neurons. And then they sort those neurons 9 days later out of the mouse brain and use the latest RNA sequencing technology, single cell sequencing, to see what gene expression changes have happened during the 9 days after the recall event in this behavior. So it combines, and I don't think we need to go into the details of how they trap the active neurons, but basically it's using these genes that they know are active, are activity dependent, that drive a reporter, and that is controlled by other ways of sort of keeping this reporter only active.
We work with this mouse line, so it's incredibly hard to explain. Yeah, cells that are on, those are tagged with a fluorophore. So the cells that were active are red in their case. Yeah, it's a funny mouse line. I think the one thing that's really interesting, that's really cool about it, is you're tagging the cells that were activated during some window, right? So it's not just defining cells by some genetic thing they express. It's what cells are active, and it gives you this kind of cool window into not just it's glutamate or GABA cells, it's the kind of ensemble, or as they call them, engrams, of neurons that are all co-activated. The one downfall of this approach is that you're labeling cells that are activated. So if you have some neural process, which we also study stuff like this, that is encoded by a reduction in activity in a specific population, you're not studying those. And so this is kind of cool because you're saying, okay, which cells are repeatedly activated and recruited, and you're kind of ignoring the stuff that's shut off. But in my opinion, kind of both of those processes, I don't think that you need to study both of them in every single study, but both of those are really, really important. I think sometimes the neuroscience field forgets that getting rid of neurons, like in development, when you have pruning, is sometimes just as important as activating things later, and kind of that yin and yang balance is such a really critical part of information encoding. But this paper is cool because it looks at what the transcriptional programs are that cause the persistent recruitment of the activated population, which is really critical for these memories.
You could look at the ones that go down, right, in a different experimental approach, right?
Yeah, it's kind of hard though, because then you'd have to have things. So we do this, and we're funded for this. You need some like double labeling approaches. So you have cells that were like activated before and now are no longer activated, which is hard. But with single cell sequencing, we're able to do that because you have they're tagging their cells with a fluorophore, but you also get the transcriptional signature of those same cells. So if you tag something 6 days ago and it's no longer activated, you should be able to tell that in the transcriptional readout of the cells based on activity markers and things like that. So with the big data in this, it's just like we have like unlimited possibilities of what we can look at and how.
The other major caveat here is that they're using one gene called FOS, or the promoter for that gene, to mark these active neurons. And there's, I would say, there's some debate on, are you capturing every single event or every single kind of active cell with this one promoter? But let's say you can capture most of them with this promoter.
Yeah. And what patterns, my husband's a scientist, and he does like 2-photon microscopy. So we've been having this, I'm going to say, dinner argument about, in these mouse lines, what activity patterns do cause transcriptional activation. So is it a single action potential? Is it repeated phasic firing over long periods of time? And kind of what is the recruitment you're getting? And so we've been talking about doing some of those kind of low-hanging fruit experiments, because we can record and tag cells at the same time, and with some of his fancy microscopy tools to see if, what patterns are being recruited. Obviously, people have shown these are involved in the memories, right? Because you lesion them, the animals don't have behavioral responses. So there's a lot of evidence these are critical. The question is just kind of in what way and what is recruitment, which is
time to do those experiments. You guys should do those experiments. I think the more of those kind of experiments that are done, the better now, because there's, like I study this one gene called ARC, which is also activity dependent, but it labels a slightly different population of cells when you look at them and you look at the overlap with FOSS. So there's subtleties here. But
that's the plan. So we have the ARC driver lines and the FOSS driver lines, and to see if different activity patterns recruit different cells in these different mouse lines, and if there's overlap and what you're really looking at, which I think is just one of those basic questions about the genetics that it's just been hard. If you don't have the tools to do it, it's not easy. So this is my fun over Christmas project.
Yeah. Sounds easy. All right. So we've got these mice. We've got these various groups that they use. So there's the untrained groups, so they don't get paired with the shock, but they see the same context. And then they have, and that's really the main control that everything, the experience that the animals are having are almost exactly the same, except they never got trained with the shock. So they're seeing the same context, and they're getting, and you can imagine that, 16 days after the first event or the first time they are exposed to this, if they're not shocked, they may actually see this as being a new context or a new thing for them, or it's novel. So you'll have some neurons that are also being lit up that would respond to the sort of newish event or experience. But the key is that then they can look at what is different between those kind of active neurons versus the active neurons during the consolidation process of this of the sort of trained animals. So that's the main comparison, is they sequence the active neurons that from these mice that were in the exposed to the context but had not been previously trained or associated with the shock, with the this group that had been trained with the shock. So that's all somewhat straightforward, but this is when, and then as Erin said, the big data kind of thing comes in, that where they have this massive data set of sequences. They sequence all the genes, and then they sort them out using what are now sort of standard computational approaches, where they can then say, okay, we have these certain cell types that are active, and they have a sort of different repertoire of gene expression, and so they sort of divide these excitatory neurons that are active into these different kinds of subtypes, and then they look to see what are the differences in the gene expression in these active neurons, or these subtypes that they've sort of defined in the in the environment, whether there's consolidation going on versus not non-consolidation. So that's figure 2, and then figure 3, they then drill down into this gene program that's induced in these active neurons that are undergoing consolidation, and they want to know, what are those genes? So what is the signature of consolidation in this population of cells? And what's kind of interesting, that's new, and I guess why Tom is particularly interested, is that a lot of the genes that are induced are involved in synaptic transmission, and especially in presynaptic vesicle release or exocytosis. And that's many of the proteins that the Sudoff lab has previously worked on. So that's somewhat new. I mean, it's all correlational here, where they're saying we worked out what the most different genes that are expressed in this population of cells are, and then we're inferring from the kinds of genes that are being induced what their function is. So that still needs to be worked out in terms of causation, what's actually going on.
Yeah. Well, part of the hard part with these activity-dependent, tagging mouse lines, is when you're doing comparisons, you may be comparing just two different like populations of neurons. And so like if you're in, let's say the nucleus accumbens, where you have dopamine that acts on one population, it increases the probability of firing, and the other population, it decreases the probability of firing. And you have something that increases dopamine, so it selects for that one population versus the other. You might just be sequencing that population versus the other, and it may not have anything to do with this kind of memory per se as it is. I think they go beyond this. So it's not just, look, this is mostly GABA, this is mostly glutamate. But I think one of the really careful things we need to do as we get into these kinds of approaches is really thinking it through, like, what are we comparing? Are we selecting for only glutamate neurons? So now what we're doing is comparing GABA, which is overwhelmingly in the one population, versus glutamate neurons, which is in the other. And then you're always going to get things coming up that are involved in synaptic transmission. And so this is such a hard question to answer, but then I think they actually did a great job because they kind of started there and that's what they identified, but then they go much, much deeper. So it's not like they got hung up on this, but it's something that I've always wondered how to get around. And I think it's easier said than done sometimes with these kinds of studies.
And one other issue.
Go ahead,
Jason.
No, yeah, I was just agreeing.
I thought that so one interesting thing to me was that they do the sorting now 9 days after the consolidation event. So the fact that the gene expression profile remains different 9 days later suggests that the cell reaches a new set point essentially in terms of its transcriptional profile, which is crazy to me, because if you think about it, this is one individual memory that's happening. These cells could be parts of engrams for many different memories, right? These cells are not allocated to this one memory and this one memory only. So it makes me it's a little bit mind-boggling. It's hard to for me to put it into what I think about in terms of what a neuron does when this single salient event can lead to long-term changes that are really independent of environment or cues or anything like that, right?
Yeah, no, that's a very good point. And of course, we have to remember this is a snapshot. So ideally, you would love to have more snapshots in between the recall and 9 days afterwards. And I'm not actually sure why or how they decided that they were going to wait 9 days per se.
I think that's to get the TD tomato expression high enough.
That's true. But, you could wait longer. Once it's up for a while. Sure. But
that might have been the earliest that they were able to get robust expression, I guess. I don't know.
Well, I would say, no, I would say you with this promoter, I mean, in a way, you're only taking, you actually are only marking the cells during that small window of reactivation during that recall event, and then you're stopping expression with the regular, with the DOX regulation. So you basically feed them on a drug that stops the expression of the TD tomatoes, because you don't want them to go back into their home cage and then re-express TD tomato in response to some other thing that they experience. So maybe they get stressed out, or they have a fight with their home cage mates. But what's that?
I was going to bring up, actually, because you just brought up the regulation. So the thing that they're using here to tag or to activate this promoter or this tool is that 4-hydroxytamoxifen. And actually, as somebody who studies sex differences, this is actually, it's difficult, right? Because all of these lines have these kind of interacting factors, right? Like DOX or this. The issue is, if you're doing sex differences stuff, is that this interferes with kind of estrogen receptor regulation. And so there's a lot of kind of, it's really hard when you're doing this isn't about that, but we do sex differences stuff sometimes, how these tools can also on their own affect transcriptional regulation in the animal. Here they have controls, right? Because their negative cells are coming from animals that also got this drug. So it's not like you can attribute all of this to that regulation, but also thinking about these tools and how that interacts with biological sex and things like that makes these genetic these genetic experiments are so complicated that it's all these interesting interacting factors. And then when you start thinking about how biological sex and hormones can influence what is regulated when, it becomes an even more difficult process.
Right. But you're right, Ori, in that so these sets of cells are tagged during the recall event, but then you're only coming back much later to sequence them, right? So this is a sustained change in their genetic programming. And I kind of think this is a really good point in that we've known for a while that there's epigenetic processes that are required for memory. And it's never been really that clear why you would need them if you're able to just have a cascade that's turned on during the event. And so this is actually in line with some other recent papers that have come out. Li Wei Sai had a Nature Neuroscience paper showing that what maybe these epigenetic processes are doing are priming the state of the cell so that they can then have this consolidation process ongoing. But then you bring up another good point, which is how do you have multiple memories, or how does a neuron participate in multiple memories?
Yeah, I think that's one thing that we have that we've talked about in a couple of these papers that we've looked at with fear conditioning is how you can have synapse specificity or in any sort of memory specificity when you have global changes in activity or transcription at the cell body, essentially. And I'm not sure that we have an answer for that.
Well, it depends what the brain region is encoding, right? You're assuming in that argument that that's the memory, as like Jason was saying in the beginning, like some sort of videotape. But what if in, for example, the accumbens, what it is it's a salience responder. So here's a great example of how that would happen is maybe you get repeatedly shocked. So now that's a really salient event. So what it does is it increases the transcriptional activity in the accumbens so that the next time you're interacting with something, it's also more salient, right? And so I think there are ways where memories, depending on what your brain region is or what the cells are actually transmitting, you could put this overlap in the kind of information that's being propagated, where maybe in some places you don't. It all depends on the brain region, the cell type. Like if it's valence, then maybe you don't get an overlap between shock and sucrose cells. But if it's salience, maybe the same cell is going to respond to both. And so I think when we think about memory in a lot of these studies, we think of memory as like
semantic memory. Yeah.
Yes. And I think it's more complicated in the brain as it's parsed into its major underlying components when you're actually using it to update information and behavioral strategies and things like that.
Yeah. And as you mentioned that the brain region where they're actually taking these cells from is the prefrontal cortex, which is I would say emerging ideas that a lot of the integration of the information coming in from the hippocampus and the sensory information about the experience, that's the site of integration is the prefrontal cortex. And that's the other thing I think that sort of speaks to your comment, Ori, is that, I can see this as being, there's a state change in the cell. So now the cell has a different genetic program and there's a whole bunch of genes that are turned on. And if a lot of them are synaptic proteins, the specificity of the memory is still coming into play here, where it's probably only specific synapses that are changing or undergoing this consolidation process. And that, of course, you're going to miss that kind of spatial specificity with this technology because it's just sequencing, total number of reads of RNA in the cell.
But that has to come from local tagging. So you can imagine that there is a lot of whatever presynaptic protein of interest you're thinking about that then gets trafficked out and maybe is only captured at the bouton that's of interest, right? So you would have in this case like a transcription modified by some local process that requires ongoing signaling.
Right. And it also kind of misses out on Jason brought up epigenetic priming. In epigenetic priming, you presumably could have a transcription factor or like a polymerase that's kind of paused at the promoter of a gene, but not actively transcribing it. And in this case, you do sequencing, all you're getting is a readout of the transcripts that are actively being transcribed. But there's many epigenetic processes that could prime something to respond when there's a salient stimulus in the environment very rapidly, faster than they would normally, that you also miss out. So, it's like one of those things that you're not going to get the holistic, you're not going to get presynaptic regulation, which I'm pumped about because I do dopamine stuff. You're not going to get these kind of epigenetic effects that are independent of maybe current transcriptional states. But it's the starting point of how we figure this out, right? And then we go and say, oh, well, are these things selected by presynaptic neuromodulators? Do we do chromatin accessibility and see if that's what's really predictive of why certain transcripts are up and not? So it's like the cool big data starting place for these more complicated questions somehow. We're in that phase of discovering the brain where everything is more complicated than the next thing.
So, one other thing about this paper that now that they sort of delve into, which I'm not exactly sure how they were able to do this with this technology. And I think this is actually, I just didn't have time to delve into the methods, but what they wanted to know was, are there transcriptional changes in other cell types outside of neurons? And they have what they say is that they again compared brains from these trained mice that underwent the recall event versus the mice that were just seeing the same environment but may not really associate that with a memory. And they see transcriptional changes, long-lasting transcriptional changes in these non-neuronal cells, the glia cells as well. And so this is, I guess, not too surprising, but what is surprising is that they're seeing gene expression changes specifically in the consolidated, the mice that are undergoing consolidation. And while there's been some hints that glia are required for making new memories, or they're modulating that process, I would say this is the first indication that you actually have gene expression changes in those cells that are required or may be required for this whole system consolidation in the cortex.
Well, I would say that they didn't show anything was required, I guess.
Well, yeah, it's all correlational. It's all correlational.
Yeah. I think the one thing that was interesting to me in this was that, I guess going back to what Erin was saying, that you could have different brain regions encoding different aspects of the memory, is that this is really a specific, this is not a general, like a state change. This is not a mouse that has undergone some like stress event that then leads to a generalized change in behavior, but that these glia are encoding some sort of, again, specific memory is really crazy.
Yeah, and I think this, so one idea is that this could be an artifact of the fact that these mice are such, they're housed in such a poor environment that this event of shocking them in a different cage and all that is like the biggest. It's like putting a 5-year-old in Disney World, and that's like their first major experience outside their home. So it's such a huge salient event that it sort of overwhelms everything in the brain. And I do always sort of feel concerned that this is not exactly how you would train that the brain is sort of just wired towards this one memory, but that obviously in a normal wild animal it's going to have lots more experiences. And so we're sort of studying these animals in a very deprived experience state.
But it also brings a couple questions. Well, two things. One thing I'm interested in is state-dependent learning and how like internal states like hunger or satiety can change how you process information. So my favorite example of this, and maybe I brought this up before, but I love it. I think it's amazing. So salt is like the best example of state-dependent reinforcement. So if you have enough salt, a really high salt solution is like off-putting. But in animals that you highly salt restrict, they are incredibly motivated to work for salt water. So you have the exact same stimulus that in some cases is aversive to the animal, but in other cases is reinforcing. And so the brain is this just like amazing system that literally, depending on your need at a given time, can change the way you process a stimulus to drive behavior. Like, how awesome. And so I think that's one that's overlooked in a lot of these studies, right?
Well, and on top of that, in many of these studies, mice or rats are already food or water deprived. Because you can't get them to perform if they're sated, right? So it's hard to compare those states because sometimes they just won't even act.
Exactly.
Yeah.
But then here's the other philosophical question I've been having with everybody in the world. So one of the things in neuroscience research, and this is every time I give a talk at an institution, I ask people what they think about this. We use a foot shock, right, as an aversive stimulus, and we use this to model aversive learning. What is a foot shock? Is it, we're not evolved to respond to electrical stimuli like that. Like, quinine and bitter tasting, we respond that way because it's predictive of something dangerous or toxic. Something really loud can damage our hearing. A foot shock is not something that we've evolved to respond to. So I have questions, and this is not a commentary in the neuroscience field. This is more of a just like thinking, what are we doing? Whether a foot shock engages the same circuits that something naturalistic would, and whether it's just such a strong salient stimulus that it just stimulates everything. We use it in my lab. This is not a commentary in the field. We also do fear conditioning.
Yeah, I'm trying to figure out though. I mean, there are, I guess there's not a lot of land animals, but there are fish that shock, there's electrical, not like it's not unheard of.
But it's just pain.
It's just pain. Yeah, that's what I was saying.
But is it pain? Because if you look up the actual definition, by the way, you can tell I've been also dinner conversation with my partner who's a scientist. It actually does not meet the definition of pain because it is not prolonged and it doesn't cause local inflammation unless it's like really, really, really high intensity. So it actually does not meet the definition of what we consider to be a painful stimulus.
But so I guess if I, if you stood on a nail, for example, right, or a pinprick, right, that's not prolonged. Like it's a quick.
But there is a physical response of your tissue to that is longer lasting than the stimulus being there. A shock is like that, and it's surprising, but this is the long story.
It seems actually pretty easy to test, right? Like just substitute shock for some other painful stimulus.
Yes, and I would urge neuroscientists, we do this in my lab, if you think something is encoding an aversive memory or consolidation, this should be true for maybe a bitter taste, a thermal stimulus. Like, is it something about the aversive experience? Is it pain? Because pain is not the same as an aversive taste, right? Those are both bad, but one is different. They are processed differently. And thinking about when we're doing these big data approaches, I think we can get a ton of information about what cells are on or off. But then when we try to go back and say, what does this circuit encode? What was this memory? How was this like consolidated in the brain? I think it's really important to think about more of a philosophical argument when you're writing your discussion about like, what really is it? And like, are we really modeling these kind of aspects of learning and memory that people
I mean, I get the same problems about fear conditioning in general, just because it's such a weird thing.
One trial learning because it's so salient. Yes.
But remember that this was all an artifact of people many years ago trying to like develop an experimentally tractable system to essentially behaviorally test the function of the amygdala, right? So think about like auditory tones. That's because there's such robust connections between auditory cortex and the amygdala. But is an auditory tone signaling danger really relevant ecologically for a mouse? Probably. I mean, maybe, but it's probably not.
Right. Well, and I think it also depends on kind of what you're studying. Like if you're in these really like subcortical or limbic structures, like maybe all of those sensory stimuli are already integrated. So a visual cue at that level and an auditory cue may overlap in a lot of circuits. But as you get up into these sensory systems, they may be different. And so this is just, you can tell this is the stuff I get excited talking about, is kind of why things were developed task-wise, what they are testing, and if they are ethologically relevant. And I mean, there's still tons of meaningful data we get from these, but there's also all kinds of fun ways we can adapt them to fit our needs in these trying times.
I have the complete opposite feel. This is almost like a philosophical discussion, and I like start to back away because I don't feel like we can ever answer these questions, especially in mice where they can't tell us what they're feeling in an explicit way.
But you can. I mean, we've been thinking about this too, and we're collaborating with a group here, Chris Gregg's group, where they've actually devised an ethologically more relevant behavioral experience for them, which is foraging. So like they have a cage where they live, and then they hook up a tunnel to a small arena where there's food buried, and mice are naturally going to go into this arena and look around, and they'll smell the food, and then they find out where the food is. And that's something, there's no punishment, there's no and it's totally taking advantage of their natural proclivity to be like, I need to go find food. And then everything's videotaped, and on top of that, the Chris Gregg's labs developed these fancy computational approaches to try and dissect out all the little bits and pieces of their behavior, which I don't quite understand. But there's a way of figuring out all this.
And I go the opposite direction. So that is like behavior entirely under the control of an animal, right? Like what the animal does. And I think that there is a ton of work that should be on that. My lab goes the other way, where we make the animal think it's controlling its environment, and it's not, because we set all the rules. And we look at reinforcement. And here's the thing. I don't have to infer what the animal feels. Did that produce reinforcement? Will an animal lever press for that? What does this drug change the rate of behavior or not? And does it matter if I change how much of the reinforcer they get? And so it's less of an inference about how the animal's feeling or what they're doing, and more of a like, this will increase the probability that a behavior will occur. And reinforcement, I love it because it's, again, under my control, even though the animal feels like it's controlling it, but it's also conserved across species. So you see the same types of behavioral tasks maintained behavior in primates and in humans. And so it's like, there's two ways you can do this, right? Go the ecological route, where we say, in this species, what circuits are involved in foraging behavior and learning? And then the other way is like, what are the conserved behaviors across species? And how do we use those to understand the brain? I mean, that's the great thing about this field, right? There's just so many ways to answer really cool questions with cool tools and behavior and all of the above. And I love it.
Jason, the experiment you were just describing, one could, the mice going into an arena and finding food, one could do the experiments described in this paper with that kind of behavior, right?
Yeah, exactly. And so we're testing this now where we have this mouse knockout where we know there's no consolidation of memory, and we run them in this task, and then Chris's algorithms can figure out it's basically a validation, a test of whether their algorithms can actually pick up what we're expecting, which is that 24 hours after the animals explored this arena, if they don't remember where the food is, they should just go into the arena and then be like, oh, well, I've never seen this arena before. I'm going to look around just like before, versus a mouse that has seen the arena, found the food in a specific spot, and then we'll go back to that spot later on because that's where they think the food is in the first place. So we'll see how that we'll see how that pans out. But exactly, so it's a more naturalistic way of doing these sort of memory tasks, but it's just not as well controlled because, as Erin says, now that the mouse can do what it wants. So maybe it's pissed off and doesn't want to like go into the cage or into the arena, or maybe it's anyway. Yeah, there's lots of differences that could happen. Whereas,
but those cool computational approaches for behavioral tracking, they really help you parse that. And then you can design experiments later. So if it's purely memory, you might see one thing. If it's motor phenotype, you might see something else. If it's spatial memory, you might see something different than if it's some sort of kind of like the animal knows there's food there. And so it's those computational models and machine learning for behavior, I think they're going to let people start to parse things that I parse really easily with reinforcement, with naturalistic behaviors. And then you go and do the second, if it's spatial memory, and then this thing should show this. If it's novel object memory, then we should be able to see this task here. And so it's just, I think that there's going to be a lot of power in those kind of approaches for understanding the brain more holistically and in a natural kind of how it functions.
Yeah, I agree. The only thing is that I just don't understand the algorithms, then I'm automatically mistrust them. But people smarter than me who have developed them can tell me if they work or not. So that's pretty much that paper. And, I think there's some interesting aspects of it, this whole sort of idea that there's the state-dependent change and that the genes that are changed are synaptic, but they're not just all synaptic proteins or a subset of ones that are regulating neurotransmitter release. So I think it'd be interesting to know, if they look at other brain regions, do you see, different kinds of state-dependent changes based on what those brain regions may be contributing to the memory? They didn't look at the hippocampus. And so, you could do this sort of comparison there where you'd expect that maybe there's a time window of this state-dependent change where we think that the hippocampus, that, the neurons that are active during the initial encoding of the memory would have a similar kind of duration of changes, and then they would drop down so they could then encode new memories. And one would predict in the cortex now that there's something similar would happen, that if you come back maybe, a month later after the recall event, that you wouldn't be able to distinguish those neurons from any other neuron in the cortex until you reactivate them with the recall event.
Jason, what do you think about these microglial genes that are encoding inflammatory cytokines and their association with this behavior? Is that surprising?
Yeah, it's not too surprising. I mean, it's kind of funny. I had this debate recently where it's, you could call these peptides cytokines only because they were discovered in the immune system first. If you discovered them in the brain first, you would say, no, these are neuromodulators, and they're they're affecting signaling, from microglia to neurons. So I think it's just a matter of nature reusing these genes for signaling, but in a different context. But then the problem is, and this is where I think the system, of course, evolution is not perfect, and since they are involved in the immune response in the disease context, now you've got all these issues with dementia and cognition, because those microglia can be activated by inflammatory responses, and now we're finding out that those microglia are actually also important for these sort of normal processes in the brain. They're not just there to react to inflammation and protect the brain.
So we must know. So when people get infectious diseases, they make IL-6, they make TNF. Does this have any impact on establishment of long-term memory? Has anyone looked?
Yeah, no. There's been a couple of papers from the Kipnis lab showing that when you have these issues with the lymphatics and the microglial activation, that you basically, the mice, when you run them on the fear conditioning tasks, they can't encode memories anymore. So there's definitely a real interplay between the same pathways that are activated during inflammation and memory. So, which is not good for us, because that's why any sort of traumatic brain injury is bad for the long term. And I think the emerging consensus is that now the microglia are playing a critical role both in mediating the protective aspect of the immune response, but also the long-term downstream bad effects, because they're now in a different state after that inflammatory trigger.
So, Jason, here we have a catalog of genes, right? Lots of genes that seem to be associated with long-term memory. So what do you do next? Do you start looking at each one by
Exactly.
itself?
So this is my huge criticism of these big data kind of papers is that it's actually, I mean, it's very easy to make a story out of this set of genes, right? Like they could say, okay, we ran these through an algorithm to say this subset of genes do these things, but is that actually true? Now you have to do the hard part and do the biology. And that's what my lab does. I sort of like rely on other people to come up with gene sets, but it's very, I wouldn't say it's easy, but of course it's fairly straightforward to get this list of genes. But then actually trying to figure out what those genes are doing is the hard part in terms of it's not very quick.
No, really it depends on what the genes are. So, I mean, like I have the same pet peeve. I think what people do, and again, I think this is out of just like how hard this question can be, is they focus on things like transcription factors, right? Which I think are great. It is really easy to overexpress a transcription factor and say, oh, things go down, behavior goes up and down. But then when you start getting these markers like snare complexes, now you're not just talking about up or down. You're talking about changing the calcium sensitivity of release regulation at a terminal. Let's say you have some sort of calcium channel. You have to know where it is and what it's doing. And these experiments to figure out what these genes are doing, that's the one side is really hard, but the almost equally hard side is to figure out why these genes, right? And so then you're talking about these complex epigenetic tools for saying, how is the nucleosome organized? Why these genes and why not those? Are there upstream regulators that are likely to have recruited this whole network of genes? Are they interacting? And, there's some really cool work. I think, Jeremy Day just had a really neat paper come out recently where they were using CRISPR. And instead of just overexpressing one gene, they basically used CRISPR to overexpress like 10 genes that they found to be regulated to see how that affected behavior, right? Because there's interactions. Yeah. And so I kind of agree. It's the first step, and you can make an easy, nice story with transcriptional profiling. It's snare stuff. Release is probably up. But then figuring out why and how is hard. Yeah.
And you hit the jackpot there, which is that up until now, we haven't had the technology to coordinate, subtle changes in gene expression in many different genes. It's always been a sledgehammer approach where you just completely take the gene out, or you downregulate the gene, or you overexpress it, or maybe you can overexpress a couple of genes at the same time, but you're not, really doing it in a coordinated way that mimics what the actual gene expression pattern changes are. And so they're always sledgehammer approach. And then the other thing is the functional readouts that you would get are very painful to do, because it's, if you want to look at changes in synaptic transmission, then you have to use classic electrophysiology. You're sticking an electrode in there and you got to record. And so it's,
and you're talking about time scales too, right? It might not be, we were doing some epigenetic stuff, and we've run into not a problem, but we're looking at histone acetylation. And there's a time window where there is an increase in histone acetylation and binding of this enzyme, but later, once acetylation is up, the enzyme is no longer bound. And so you're also looking at the time course of this and how fast it happens and how that relates. And so it's also not as easy as saying at 9 days. I mean, this is great because they were able to do that. But for many things, there's a difference between 30 minutes and 2 hours and 4 hours. And I'm excited with CRISPR and all these really cool tools, especially on the single cell genomic side and the big data approaches to behavior. I think that we're getting more data, but now the next thing is how to use that data in a theoretical framework to solve how the brain works, rather than just dumping it out there and saying, there it is. And I think that there are people thinking about that, but it is the hard part.
It's easy to just, and that's why we every second paper that's coming out in Nature and Science right now, these big data sets where it's the new revolution of single cell sequencing, and you can come up with a whole bunch of sets of different lists. But now what? That's the big question. Is how do you put that in a framework that's testable and functional relevance is the hard part.
And these lists of what these proteins are doing are also coming, and you know this better than anyone, they're coming from lists of what the proteins were studied in. So when you see things that they say, oh, it's this, and so you say, oh, well, it's obviously involved in olfaction because that receptor was involved in olfaction. But then you discover actually that protein that you thought did only this is actually doing something very different in this system. But in your O-term list, it says it's X, Y, and Z. And so I think we're also, I don't want to say being led astray, but I think if you take the simplistic view of how these big kind of data pipelines work, I think we're missing out on a lot of the really cool data in there when we're not thinking about it from a biological approach of like, does this even make it to protein? That's like the first step.
Exactly. And that's why like that last paper we discussed with that orphan receptor that came out of the screen that was completely unbiased. That was an amazing set of experiments and a ton of work and forward genetics and whatnot. But the outcome of that was unexpected in that you had now identified this receptor that was, totally not studied before. And with this approach that we were talking about, where sort of the gene ontology is based on all the basic science that people have done, if there's nothing known about that gene, you're going to completely ignore it in these lists.
Yeah. I'm so glad you mentioned that, Erin, because this is a transcriptional analysis. We don't even know if they're made into protein, right? And there could be post-translational changes that have an effect as well.
My background is transporter regulation and function, and most of its regulation is at the post-translational level. And so it's you're just saying up or down is not enough to predict its function. So I went to a genetics lab for a postdoc from the transporter field. So I think I have a maybe unique perspective on, does this actually make it to protein? What else?
I think that's important. It's important.
Yeah,
Many people do.
Arguments with people here.
Yeah, I mean, if you do these big transcriptional analyses, you don't think about that because you can't. You have to, and this is what you're selling. And same thing in virology. You would get these listy papers, and then nobody ever comes back to it. It's really big, prominent journal, and that's the end of it. And what's worse, you can get a listy thing in 3 different labs, and the lists are 3 different lists, right?
Yeah.
That's the other thing about big data sets. And I think one thing we didn't touch on is in single cell sequencing, what you do is this clustering analysis to try to identify groups of like cells. But here's the problem, that if you don't have thousands and thousands of cells, as you increase your cell number, the number of clusters you identify also increases linearly. And so if you see a paper and they say, we found 6 clusters, if they sequence 3 times as many cells, they might find 18 clusters. And so, it's kind of, I think that we just have to be careful with these approaches, not because I don't think they're giving us information, but using the theory of what we've done before and what we can do after to really put them into a framework of how the brain works, right? This paper doesn't exist in isolation. There's tons of work on engrams and other brain regions, and you think about how that fits. And then I think it's super powerful.
Right.
And these are also dissociated cells that are getting FACS sorted. So who knows whether these transcriptional changes are actually happening in vivo, but.
Yeah. And in fact, there's a whole bunch of RNA that's being trafficked out into those synapses as well.
Right.
And the process is that that's actually my other big pet peeve about single cell sequencing in neurons is that we know that there's this whole repertoire of RNAs that will totally be missed because you're only taking the cell body and you're taking off all the other bits and pieces of the neuron. Or just the nucleus, yeah.
Well, the nucleus is more fun because you can do that from frozen tissue, so I'm sure it's fine.
So that's a good place to end. Even though these techniques that enable this are fantastic, they still have limitations, right?
For sure, definitely.
And of course, these are mice, so that's another layer. And maybe if they did it in a different strain, you'd get different results, right? From the strain that was used here.
Right.
Okay.
Yeah.
Thank you, Jason. That was great.
Thank you.
All right. That's TWiN 13. Show notes at microbe.tv/TWiN, TWiN. Questions and comments, TWiN at microbe.tv. If you like what we do, consider supporting us. Go to microbe.tv/contribute. Ori Lieberman is usually at Columbia University Irving Medical Center. He is at Ori Lieberman on Twitter. Thanks, Ori.
Thanks, Vincent. Thanks, guys.
Erin Calipari is at Vanderbilt University on the Twitter, the Erin Calipari. Thanks, Erin.
Thanks.
Jason Shepard's at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent. So we didn't get any emails again, huh?
We have a big one there, but I thought we'd leave it till next time because
Sounds good.
This was a great discussion on its own and doesn't need to be distracted from. That's what I thought. Plus, it's pretty late here.
But we should get, exactly. But we should get, listeners, when you're listening, send us some paper ideas as well. I think it's always fun to know what you all, are interested in versus us and cherry picking our papers here.
I think in the realm of science right now, all attention is on this virus, right? And as soon as we can get away from that, it'll be better. Because even neuroscientists have turned to COVID-19 because it's doing things in the CNS, right? So
there's a big Ed Young article in The Atlantic about that, about how science kind of turned and how there were good things and bad things that came out of the last year of refocusing in science.
Yeah. Oh, totally.
Amazing amount of people did switch completely, and now it's amazing to see how they switch back.
So I got to be a little cynical here. Despite all this, we have neuroscientists coming in, we have immunologists coming in. In the end, you just make a vaccine, and that's going to be the end of it. And they started the vaccine back in February before we had any knowledge of the immune response or anything else about COVID-19. It just is empirical vaccinology. So, we're going to learn other fundamental stuff, but none of that, you could argue that all the untold studies on how long the immune response lasts, how durable is it, doesn't matter as long as you have a vaccine that does it, right? So
ultimately, I totally agree. I wonder if this is going to be turning into what happened with HIV, which is, in the end, the way they justify the research is that we are learning the basic components of how all of this works, and maybe that'll speed up vaccine, making better vaccines. But if the vaccine works at 95% already, it is what it is.
I mean, I would argue that we really haven't learned anything new fundamentally about infectious diseases and virus reproduction from studying SARS-CoV-2. Okay. Everything that has happened is built on what we know from other viruses. And it's a weird disease. It affects a lot of organ systems, but we still don't know the basis, the pathophysiological basis for any of that. And so, I mean, I'm not saying we don't do this, but the way everyone has just jumped on this and, they justify it, we have to do this. We have to work on it. It's like, really, you do? Why? They're making vaccines. They're making antivirals.
I decided early on that we weren't going to do it. I mean, there was a time where the lab was shut down, and the only way you could do any research is if you said, I'm going to do COVID research. And so many people did say, I'm going to do COVID research. They could at least have something going on in the lab. But I felt like, we weren't going to make a big impact on the therapeutic side. What was the point? But
there's also some psychological aspect of, we're all sitting at home. We want to be able to help. So there's at least we're doing something.
I understand. But I got to say, for the first 3 months, all we had was list papers. Let's infect cells and sequence all the RNAs that get induced, right?
That's true.
Really? What did you learn from that? That the vaccine is going to... Anyway, I mean, I'm all for basic research because that's what made the vaccine go so quickly. But I just don't like this herd mentality that everyone runs to it and happens every time there's an outbreak, right? It happened with Zika. It happened with Ebola. Sorry, Erin, what did you say?
No, I was saying the same thing happens in my little addiction field too, right? It's new drugs crop up. Like there was bath salts for a minute. And then all of a sudden people are changing their research programs to bath salts, which is great, right? We do need research on that. And people were studying it before, but then all of a sudden it's now the new problem and the new problem. And I think sometimes when you do that, you're making less of an impact than actually steadily studying something, building work over time, and maybe letting the academics do the work.
All right. I'm Vincent Racaniello. You can find me at virology.blog. And you have been listening to This Week in Neuroscience. Thanks for joining us, and we'll see you next time.